Your show, Alex.
Awesome. All right. Okay. So my name is Alex Martin, and I will be talking about my master's project, which involves this aquatic plant genus Vallisneria. So before I talk directly about Vallisneria, or Vals for short, I want to briefly introduce aquatic plants in general. So what makes a plant an aquatic plant? A basic definition is that the plant lives most or all of its life history in or around water. And we can break this down into about 4 categories. The first being plants that grow along the shoreline of lakes and wetlands, or those that are in the riparian zone of a river where the roots are constantly saturated by water. An example here is Lobelia Cardinalis. It's a Kansas native hummingbird adapted species. It's quite popular among gardeners, but some people don't know that it's been a staple in the aquatic plant hobby for a long time. Lobelia on the left is being grown completely submerged in water, and it does so quite easily. And one of the issues that I'll touch on later in this talk concerns the differences that you see here between the way these plants look based on their different growing conditions. The second category for aquatics is emergent plants. This is an example here from Kansas as well. It's an arrowhead plant. So the roots and some portion of the stems are underwater, but the leaves and the flowers stay above the surface. The picture on the right I took at the KU field station at one of our research ponds. The third type is floating plants. This includes water lilies on the left, and then on the right you can see some truly floating plants like water lettuce, water hyacinth, and the small ferns called salvinia. These 3 are truly floating, meaning that they are at the mercy of wind and currents of the water to move them around like a lake or stream, as their roots don't ever anchor into anything. And then the fourth and final type of aquatic plants are those that are submerged and live entirely underwater. And the example here I'm giving is Vallisneria, the topic of today's presentation. And aside from the flowers that they produce, every part of the plant has to remain underwater in order for them to survive. So Vallisneria are an aquatic monocot, and the generalized body plan of these plants can be seen on the left. They grow leaves from a rosette or crown at the base of the plant. The roots anchor the plant into the substrate, and then they also produce clonal runners called stoloniferous growth. These plants are dioecious, meaning that a plant is either male or female. The picture in the middle shows a male plant that's producing these spathes that are basically like hermetically sealed balloons containing hundreds of tiny male flowers. And once they mature, that spathe opens up and those male flowers get released to the surface of the water. On the right, you can see that I'm holding some female flowers. Vals will produce a single female flower along a long stalk called a peduncle that will float at the surface of the water when they open up. Here's a picture of a female flower that's surrounded by a dozen or so male flowers. These plants are entirely water pollinated, and the white portion that you see in the female flower is actually the stigmatic surface, and that's what's receptive to the pollen from the male flowers. Here's some color variations of the female flowers that I have noticed in my collection that I have growing here in growing at campus on my home right now. But going left to right is Vallisneria americana, which is found here in the United States, and then Vallisneria spiralis from Europe, Vallisneria gracilis, and then Vallisneria nana, which are both from Australia. And one reason that we might be seeing these coloration differences and like this red pigment that's being made that's called anthocyanin is it could be a response to different sunlight exposures, but we would have to do some sort of additional study to verify this. So here's some microscopic images of the male flowers that I was telling you about earlier. On the left, you can see I pulled an immature male flower out of one of the spathes. I cracked it open and pulled these out. You can see that the tiny little yellow dots that are inside there are actually the pollen grains. And this little balloon, once it gets released from the spathe, it'll float up to the surface of the water and crack open. And then that 1 or 2 cell layer thick sepal will reflex and act like a flotation device for the flowers so that they can move around the surface of the water and then eventually come into contact with the female flower. And just kind of an idea of how small these things are, the far right picture, that's the tip of a pin. So these flowers are like maybe 1 millimeter tall. And then I talked about like the basic body plan of what makes up a val, but there are some exceptions to this rule. Here's 3 species that are found in Australia, where the leaves grow along a stem rather than just at that base crown. Going left to right is Vallisneria caulescens, Vallisneria triptera, and then Vallisneria rubra. In 2008, Vallisneria rubra was removed from the monotypic genus Maedenia and then transferred into Vallisneria to maintain a monophyletic group. And then here are some pictures to show the different variation in leaf morphology. You can see that there's some major differences in the coloration patterning, and all of which remain consistent even when they're all grown in the same environment. So on the right, the far right picture, you can see that I'm showing that one of the features of vals is this serrated leaf edge. Some species, they have this serration going all the way down the leaf, and then some of them just have that serration right at the very tip. Vallisneria are found worldwide. So here's a map that I pulled from GBIF, which shows the occurrences from both iNaturalist and from herbarium specimens that were cataloged with Kew Gardens. The genus contains 14 valid species for now. So I'm hoping that my research will shed some light into some of the taxonomic issues that are currently in place here. Some previous systematics research includes the 1982 paper from Richard Loden. He was one of the first to attempt a full synthesis of all the plants in the genus, but he noted that there were many difficulties associated with vegetative morphological features. So a lot of his analyses used mainly floral traits. And in 2008, Les et al. Came out, which became the most robust investigation into the genus. They were able to analyze both molecular and morphological data from all available species at the time, and then produce the group's first phylogeny. So all of the tree building here was done using parsimony. And so we can see on the tree on the left, using just the molecular data, there's some polytomies and some low resolution clades found throughout the group. The addition of morphological features seen on the right resolved some of these issues, but there was still some problematic areas in the tree. And with my past history of growing aquatics as a hobby and the data that I was collecting with the living collection on campus, the one concern that I had using some of these morphological features in tree building was something called phenotypic plasticity. So the plants shown in these 2 pictures are the exact same plant. They're just grown in 2 different conditions. The plant response to different environments can be, it can attribute to like discrepancies within morphological data. So like if you're using metrics like leaf length or leaf width, for plants that have been collected, you're not really taking into account the variation that you might see in these species when they're grown in from one condition to another. And so I wanted to dispel some of these issues by having all of the plants grown in a common environment, and then measuring those features and then placing them within the matrix that was done in Les et al. 2008. But I had a lot of issues when I was measuring and placing these measurements into the character states, because they were either like ambiguous states where it was kind of like your choice on what you decided, or when I did measure them, those measurements didn't fall within those character state categories. So with that in mind, my master's project was developing with the help of Mark Mort, Lena Heilman, and John Kelly. And I was able to come up with some research objectives for this project. I wanted to get all 14 valid species sent here to Kansas, verify the work from Les et al. 2008, ask Don to be on my committee to help with the project, and possibly get the DNA that he had stored at the University of Connecticut. And I wanted to look into finding some additional gene regions to sequence, and then produce a new phylogeny using maximum likelihood. Unfortunately, I started my master's at the height of the pandemic, so a lot of these things got taken out pretty much immediately. Missouri Botanical Gardens closed itself to all research. There was really no possibility of obtaining plant materials. All shipping and importing was like cut off from everywhere, especially from Asian countries. Don did agree to be on my committee despite his retirement status from academia, and he even made the trip to the University of Connecticut but was greeted with the -80 freezer that was broken, and it had a sealed, it was also sealed shut due to a faulty lock. And once they finally got the freezer open, they were greeted with ruined tissue and thawed DNA. So everything that he had collected for that past research was toast. This immediately knocked out the possibility of adding in new genes to the data. I could use GenBank sequences to produce a new phylogeny, but I didn't have like any additional plants. So I turned to the internet. I made posts on forums that I had been associated with in the past, made posts on Facebook and Reddit, and I was asking people for like help with this project. And a common name that kept popping up was this guy. This is Tom Barr. He's very well known within the planted aquarium community. He has a PhD in aquatics, and he runs his own website that's dedicated to learning about aquatic plants. I got in contact with him, and he let me know that there was a possibility of getting some plants out of Australia, so he directed me towards his longtime friend, Dave Wilson. So Dave is a small business owner. He's just outside of Darwin, Northern Territory, Australia. And for many years, he's been curating and selling Australian native aquatic plants and fish. He's the go-to guy for researchers looking at field collecting in Northern Territory. He regularly shows these researchers where to find certain plants and fish, and even voucher specimens with the Darwin herbarium from collection trips. And without the help from Dave, this project never would have happened. He maintains a living collection of almost everything he's collected and keeps amazing records, including dates, locations, and which researchers were there at the time with him. He agreed to send me plants and noted that one of them that he wanted to send me was possibly a new species. So he had done a lot of work with his friend Surrey Jacobs. He was a researcher out of the Sydney Royal Botanic Gardens who had published like he published many papers with Don Less. And unfortunately, Surrey died in 2009 due to cancer. But at some point before his death, he thought that the one particular plant that Dave wanted to send me, that I'll talk a little bit about later, could be a new species that hadn't been described yet. And so Dave asked me that, if my research did show that it was a new species, could I name it after Surrey? And I wholeheartedly agreed, like, yes, I'll do this. And so, Surrey was an amazing scientist who did tremendous work for aquatic plants in Australia. But now the issue that we faced was how to get these plants here to the United States alive and in a legal manner. So I had previous experience with exporting plants while I worked in Lena Heilman's lab. And the USDA made it fairly simple. You make an online profile, fill out some paperwork, and then the next day a person came to campus, inspected the plants, gave us instructions how to ship them out, printed off a phytosanitary certificate, and we were good to go. We had the plants ready to go. The next day they were off to Canada. So all in all, it was like less than a week for the process. So the Australian government has this listed right here as the 9 steps needed to, export plants from their country. They made it seem fairly simple, but it literally could not be any further from the truth. First of all, Vallisneria was not on their list of either prohibited or non-prohibited plants. And when I let them know about this, they told me I was lying or I just missed it. And then like a week later, they emailed me and said, you're right, vals are not on the list. So we're going to form a committee and decide from there. And so they got back to me another week later and said, vals are considered non-prohibited as they're very common in Australia. And so next thing I had to do is get Dave Wilson registered as an official exporter. And then here's where it gets a little bit more complicated. Unlike the USDA, who just sends you documents directly, the Australian government requires that you hire a middleman company to access their electronic document software. So they send the documents to the company sends the documents to me, I fill them out, send them back to the company, and then they import them back into the software. It's like a giant scam that adds in more steps than what's actually needed. And once I had all the paperwork done, like once I was finished with all this, we had to get the plants inspected by an authorized inspector. The Australian government refused to give me a list of people in Darwin that Dave could go to because they feared that the list would favor like one inspector over the other based on how they sorted the list. But we did end up going with an inspector that was from the Australian government, and it was basically a shakedown. They charged $75 every 15 minutes to inspect the plants, And it took them over an hour to inspect a dozen tiny, tiny little plants. And so I just want to thank Dave for his perseverance through this whole process. And I want to thank Eric Anderson from the USDA for the help. The plants never would have made it here alive had it not been for both of them. So I want to thank them. And so I was able to do my own collecting for this project. I went back to Minnesota for the 4th of July last summer, and I collected some Vallisneria from here at Square Lake. It's just north of the Twin Cities. It's right near the border to Wisconsin. This lake is known for having very clear water and visibility up to about 30 feet. So there were some previous collections that were here with Vallisneria from like different herbariums around Minnesota, but nothing that had been collected recently. So I went out onto the lake with my cousin PJ on his boat, and while I had my arm hanging off the side, I was hanging off the side of the boat with like my legs under the water and my head sticking down with the swimming goggles on. We trolled around the edges of the lake looking for plants. Anytime I'd see some, I'd tell them to stop. I'd dive down, grab them, and collect the plants. So here's kind of what the plants look like in the bag. We went around the lake a few times. I was actually looking for evidence of female flowers, so those flowers resting at the surface of the water, but we didn't see any. July may have been a little too early in the season for them. And all of the plants I ended up collecting ended up being male, so I don't know what's happening there. Well, I also wanted to collect plants in Central or South Florida as well, because there is currently one species listed for all of America, and that's Vallisneria americana, but there's some debate on this issue. And so my wife and I decided that we wanted to make this a dual purpose trip. We were celebrating our 10-year wedding anniversary. And so we flew into Orlando and then drove west to Hernando and stayed at an Airbnb. Our cabin was located on Lake Hernando. And to my amazement, Vallisneria was growing right there off the dock. It was awesome. It's the first time I had ever been able to see Florida native vals. And I immediately saw that there's a difference between these and then the ones that I collected in Minnesota the month earlier. The Minnesota vals are entirely green, and they have no striation patterns, whereas these are much more colorful and they have incredibly large leaves. So I had a particular collection site in mind before we went there that somebody had posted on iNaturalist, which is this location here. This is Little Jones Creek. It's just north of Lake Panasoffkee. It's a spring-fed creek, and it maintains a temperature of around 72 degrees Fahrenheit or 22 degrees Celsius all year long. The vals here are ginormous. So they're like 8 feet long. The leaves are like 8 feet long, like 2 inches wide. They're huge. And I struggled to go in and collect plants that were small enough to fit in that gallon bag that I was using for collecting. And so Renee and I were looking for some activities that we could do while we were in Florida for our anniversary. And she came across this clear kayaking tour that was on the Crystal River, which is located on the Gulf Coast at Kings Bay. And so here we are in the kayak at the Three Sisters Springs, and that feeds millions and millions of gallons of fresh water that mixes with the salt water from the ocean and creates this brackish environment in the bay. And so right when we arrived at the docks for the tour, I noticed that this whole bay was like covered in Vallisneria. So here's a picture that I took from the kayak. Like you can see, you can't see the sand at the bottom at all. It's completely covered in vals. And the tour guide told us that there was this restoration project that was planting these vals to maintain a healthy ecosystem for the manatees that would come in during the winter. And at some point, a huge storm pushed in a bunch of salt water and killed off all the vals that were there before. And there was one thing I noticed. I was like, these vals that are here right now, they look really similar to the ones that were in Minnesota. And so they look different from the ones that I saw in Hernando and then Lake or Little Jones Creek. So I collected some for the project. And with that, I was able to, with the new plants from Australia and those that I collected from Minnesota and Florida, I was able to, my project had more depth to it. I wanted to see if the one plant that Dave sent me was a new undescribed species, resolve the taxonomic issues that, there might be here in the United States with the Florida native Vallisneria and then the ones that are part of the rest of the US, and also see if there's some sort of parallel that might be happening in Australia as well. So I got working on DNA extractions with the help of this device on the left. We call it the D-annihilator. I think it was coined by either Ben Curbs or Keeley at some point. So after I got the DNA, I ordered some PCR primers for the 3 gene regions that were previously used in the Les et al. 2008 project, and then played this fun game of, are my PCR products amplified enough to send in? So like the picture you see on the right, like, do I send in all those products, or do I like redo some of the ones with the faint bands? A lot of trial and error, but eventually we got all of our sequences sent back. They were in text format and chromatogram data. And so I visually checked every single chromatogram front to back using Chromos, and then I assembled using CView. And you'll see here that some of the sequences had single nucleotide polymorphisms. And so from previous research, we know that double peaking in this ITS region has been suggested to be a result of hybridization. And so I included all the hybrids in my initial analyses, but one of them was subsequently removed from tree making because it kept bouncing around to different locations on the tree. So I used IQ-Tree for tree building using maximum likelihood. I tried different iterations of partitioning the 3 gene regions, but the logs kept getting spit back to me saying that no partitioning and using one model of evolution was the best route. And so this tree was produced using both Les et al. 2008 sequences, plus the sequences from my plant collection on campus. And mine are the ones that are highlighted. And then I will touch on the one that's in red towards the bottom in a little bit on this talk. And so these are kind of questions for you that I'm hoping that maybe you'll ask or help me out with later. I'm not sure what else can be done with this data. You know, we're pretty limited on parsimony informative sites, so I'm not really sure if there's any other methods that could be used to resolve this tree any further. The topology between this tree and the one that Don Les did in 2008 is nearly identical, except for the placement of Vallisneria caulescens, but even that has a weak bootstrap support. And then one thing that Les et al. 2008 noticed was the lack of resolution in this Vallisneria gracilis, Vallisneria innua, and Vallisneria erecta clade using just molecular data. And this held true as the addition of any other taxa to this group had inconsistent placements with the species. So like same species didn't match up with one another. And as at the moment, it's odd, Vallisneria gracilis is not a valid name. It's actually a synonym with Vallisneria nana. So there is an issue like in this area of the tree for sure. But I did, however, see that the one possible new species of plant here that I have labeled as Jacob's Val, and then the 2 others that were collected in the Northern Territory formed their own clade that was sister to like the rest of Vallisneria australis. It's interesting because the name australis means from the south, which this plant was thought to only be located in southern Australia, so like New South Wales, Melbourne area. So there appears to be some sort of parallel happening in both the United States and Australia, where we have these monophyletic groups splitting themselves up internally based on climate. And so the current classification of these species, it lumps both of those together to maintain the current naming system. But an argument could be made that these should be split up into separate species or at least different subspecies. So here's a look at the possible new species. It's unlike anything I've seen so far in person or in literature. It has this amazing coloration pattern with bright yellow-green stripe going right up the middle of the leaves and this vibrant red color. It has these dark red striations. And it was right about the time that I was looking at this plant that I had ordered a book called Aquarium Plants by Christelle Castleman. And I was paging through a lot of the plants that she had in the book that were a part of the family Hydrocharitaceae that includes Vallisneria. And then I came upon this plant right here. So I kind of had like a little mini heart attack when I saw this. This is Odelia brasiliensis. And this plant looks remarkably like the Val that Dave had sent me. And like the coloration pattern was almost identical. Both plants are from the same family, but they're not closely related to one another within the family. and they're not even located on the same continent. This one's in Brazil, the one that Dave sent me. It came from here in Northern Territory. It was collected in an oxbow lake, or what the Australians call a billabong. That's a lake that's left over from the meandering of a river, and then it eventually gets cut off at some point, and that lake becomes isolated from the rest of the river system. So I thought that was really interesting when I saw that. And during my time that I was collecting the permit for Florida, I was emailed by Dr. Gorham, who told me she was also working on Vallisneria. We had a Zoom meeting where we talked about our research. She was looking into the presence of hybrids that were popping up all over Florida. And at the time of our meeting, I was still getting sequences back, and I didn't have anything concrete yet, but her description of what the hybrids looked like, it sounded really familiar. And I did a little bit more digging into this topic. And you remember the valves that I said I mentioned later, the ones highlighted in red right there? So that was the plant that I collected from that clear kayaking tour. So those plants were from that big restoration project, and the people heading the project, this Kings Bay Restoration Project right here, they claim that they found a saltwater tolerant Florida native val to replace the ones that were lost in the bay. They hired a nursery company to grow this giant stock of vals. They worked on removing the cyanobacteria and harmful algae that was covering and choking out the bottom sand on the bay. And then once that was all cleared out, they transplanted the Vallisneria. And so I collected those vals, and then I got the sequence data back. I tested that with Dr. Gorham's tree that she had built using the ITS sequences. And yeah, it turns out that there is very strong support to suggest that the vals that they're using for this project are the exact non-native hybrids that Dr. Gorham and her team have been finding popping up all over Florida. And so I can only suggest at this point and guess, but I think that this restoration project found some vals growing in an area that were saltwater tolerant, and they assumed that they were native. And then when they used them in this project, they gave them these catchy names like Rockstar and Salty Dog. And I'm assuming that they don't know that they're using hybrids, but either way, this is bad. And so when I figured this out, this was basically the face I made. This is the lead singer to a band called Trivium. He actually lives in Orlando, Florida. His name's Matt Hafey, so I thought it was kind of fitting here. And so yeah, even the science is not immune to the Florida man issues that are being faced down there. So with that, I was able to add in a couple more objectives to my research. As you can see, I've stated down there below. And if this project ever goes into like a PhD or any other type of project that I want to work on, if I want to keep going forward with this, I would like to try to get some genome level sequencing. If that's not possible, find some better regions to help resolve those Australian clades. Add in the missing taxa that I wasn't able to get from China or the other Asian countries where they had, newer taxa that were described like right at about the same time that Don Less paper came out. And to this last point, so we know that waterfowl here in the United States, like the canvasback duck, are known to eat Vallisneria plants. It's actually why they were named their species name. And there's also observations out of Australia where they're seeing birds eating Vallisneria fruits. So there might be something happening where we have these migrations of birds who are eating these fruits across a country who are laying these seeds in different pockets of water, which is why we might be seeing, Vallisneria australis that's typically found in the south starting to pop up in those tropical areas. So with that, I'd like to thank you for your time, and I will answer any questions that you might have.